2 for nutrition. This is ecology and growth, looking at these concepts of alterations with growth based on environmental factors. And then the concept we've already covered we've already covered both of these in the laboratory, but microbial growth, looking at both individual cell growth, but specifically population growth factors or population growth. The last 2 key concepts, remember we had 2 already. One was tonicity, or looking at osmotic pressure, and the second was understanding passive versus active transport. The last 2 key concepts for this chapter are understanding oxygen processing and looking at that bacterial growth curve and understanding what's happening at each stage along the way. Now for oxygen processing, I will forewarn you that this concept is going to bleed immediately right over into our chapter on metabolism, where we're going to use these terms when we get into metabolism and look at specific metabolic pathways that are associated with each of these oxygen processing terms. But let's start with the easy stuff. This one we're not going to spend a ton of time on. I think these are just basic terms for you guys to go through and memorize. So as with any organism, organisms are susceptible to their particular environment. For bacteria especially, they are very prone to alterations with any changes in their environmental factors like temperature, oxygen, pH, etc. There's a whole bunch of other things on this list. Salinity we'll talk about too, because they really have very little homeostatic mechanisms, right? Ways to internally regulate these different factors, and therefore they essentially take on their ambient environment. So whatever the environment is, that's pretty much what they are too. Now I'll also just put a little side note in here that everything we're talking about for growth factors or things that change, environmental factors that will change the growth rate of organisms, just keep in mind that it's all based on enzyme function. The reason that bacteria die under high temperatures is because their enzymes stop working. They're denatured. The reason that pH messes them up so much is because the high proton or high hydroxy ion concentrations will interact with enzymes and cause alterations to their function. Now, there are, of course, other macromolecules in the cell that could be altered and damaged in any of these environments as well, and that does happen, but the primary reason that you see altered growth rates when you have changes in temperature or pH or any of these other factors is due to enzyme activity more so than anything else. All right, so the terms here are used to describe the range versus the best condition under which organisms can grow. Now, your textbook really only uses minimum and maximum and optimum, these terms here, when used to describe temperature conditions. But I, of course, I like to use them for really any condition. But again, just understanding how these terms are used, these terms min and max are used to describe the range. That means the span of conditions under which an organism can grow, versus optimum is used to describe the best possible condition. That will just be kind of one spot along the way, and again based on enzyme activity. But I think a graph would be a much better way to understand these. These are terms that you should know, right? So memorize these terms here. And again, this is looking at these 2 terms, minimum, maximum, and optimum or optimal conditions. So, for example, these psychrophiles, an organism that can grow right between negative 20 degrees Celsius and 15 degrees Celsius, which is very cold. These guys, this is their range, their minimum condition, their maximum condition that you could still see growth. But then their optimal, where they grow the best, is between 0 and 10 degrees Celsius, right? So you can see these ranges, min, max, and optimal for all of these different organisms. But again, to visualize them, I think is much better. We've seen this already in the laboratory. You can pull this right out of your textbook. And again, for those of you who are more visual learners, this is exactly the same information, just put graphically. So let's look again at those psychrophiles. And if you need help figuring out the difference between these, remember, phile means loving, kind of, right? Troph means growth conditions, but phile means loving. So whenever you're trying to remember which is which, just like think of psychro as cold. Psychrophiles like really love the cold, so they're way down here, versus psychotroph. They can tolerate the cold, but it's not as much as these guys up here. Okay, I mean that's just one way to think of it. All right, so these blue guys are these psychrophiles. We just saw again that their range is between negative 20 and about 15 degrees Celsius. So if you were to plot this out, you would still be able to see growth all the way down to this minimum condition. You could see growth all the way up to the maximum condition, but you can see the optimal range here, or the optimal condition, is between 0 and 10 degrees Celsius. And again, this would be completely based on enzyme activity. So where their enzymes function optimally is between 0 and 10 degrees Celsius. So if an organism performs like this and you can determine it, you don't have to go to enzyme activity to determine optimal. Really, you're just looking at population growth. So just like the lab we've performed, where you can have a test tube that's put in a very specific temperature condition. If you see growth in that temperature condition and you measure growth at other conditions, then you can kind of plot out their population growth and determine their temperature preferences. So again, range is based on just these numbers down here. Optimum is based on the rate of growth way up here, right? So this term is optimum, and these numbers down here would be the range. So you can see that these guys overlap, right? So if you were just to have one test tube at 10 degrees Celsius, you saw growth in it, you can't tell me if you're working with a psychrophile, a psychotroph, or a mesophile, because all of them have growth at 10 degrees Celsius, roughly speaking. But you would have to then establish their ability to grow at different temperatures, so therefore to then decide which characterization to provide for them. Now, a note here, cold does not usually kill organisms. It very, very rarely does. Usually cold is actually used, in the laboratory we use cold to stop the growth and actually keep the organisms in a dormant stage for an indefinite period of time. Negative 20 degrees Celsius, that's like your freezer. Negative 80 degrees Celsius, that's getting a lot colder than that. That's a deep freezer, and then liquid nitrogen is much colder than that. That can really help preserve these organisms in this dormant stage. So when you are thinking this in application, if you have food that has been prepared, that means it's been exposed to microorganisms, good or not good, but let's say that there are potential pathogens on there, and you put that food in refrigeration, that's good because you stop the growth of microorganisms. But just note that once that food comes back out of refrigeration and is set at room temperature, the organisms are not dead. Therefore, as soon as that food reaches ambient temperatures of the environment, the organisms will start growing again. So keep that in mind next time you set your chicken out on the counter to defrost and you sort of just leave it there all day. Well, as soon as that chicken has gotten up to room temperature, that salmonella is definitely going to start growing on that piece of chicken. For disease, just consider your body. Remember, we talked about this in the first week of class. Your body hosts a wide range of organisms due to its different habitats, and temperature is a key factor here. So you'll definitely have different normal flora at different parts of your body, and also different pathogens in different regions of your body. This is a great discussion question, and one that you will find just like this on the exam. So I really like these application-based questions. Cultures of bacterial species were incubated on the shelf of a refrigerator about 10 degrees Celsius, out on a lab bench, at the bench top of a lab at 21 or 25 degrees. It's all very similar. On the shelf of a 37-degree incubator and on the shelf of a 50 degrees Celsius incubator. After incubation, there was no growth at 37, no growth at 50, abundant growth out on the benchtop, and slight growth in refrigeration. What term can be used for this species? Well, to help answer this question, let's draw this out, as I will do this question with you. So if you were to plot out, just like we had up there, the temperature range where we have 10 degrees here, we have 21 degrees, 25 degrees, 37 degrees. I know I'm not making this perfectly linear, but you get the picture. 50 degrees, and we said there was no growth here, there was no growth here, there was abundant growth at the benchtop and slight growth at refrigeration. So a whole bunch of growth here and a little bit of growth here. Again, if we were just to kind of plot this out, and then, whoops, that should be down to 0 because we said no growth there, then we'd be really looking at this number here, right? Wherever it said optimal growth, this is what's going to give us our term. So then we just have to go back to our graph and find where do we see optimal growth at 21 degrees Celsius. Well, here we go. It's a psychotroph. And you can use the range we just determined to see if this fits. So do these psychrotrophs grow at 37? No. Do they grow at 50? No. Can they grow at 10 degrees? Yes, but very poorly. Does that fit with our scenario that we've just described? Yes. Oops, we're already here, right? That fits perfectly. So when you're doing any of these practice questions or see something like this on the exam, I know there's a lot of words here, but I find it very helpful to draw out something like a graph and then really focus on these terms, key terms, abundant growth, right? When you see abundant growth, we are talking about its optimal conditions, and therefore we are talking about, or therefore you can give a characterization based on the terms we've just learned. Some of these other environmental factors, we just looked at temperature, and now let's discuss pH and salinity. So for pH, there's only a few terms that you really need to know. Most organisms do prefer a pH between 6 and 8, which is roughly in that neutral range. You can call them neutrophiles if you want a term for that. Those that prefer pH between 0 and 2 are considered acidophiles. Remember, just to keep this in mind, that term philes means like loving, so they really prefer that environment. Alkalinophiles or alkalophiles are organisms that prefer between 12 and 14, but you can keep it as simple as just saying anything above pH of 10, where they have their optimal growth. And again, those are optimal conditions. This is a picture of an acid mine that has really destroyed the ecosystem in this particular region. And actually this kind of refers back to something that we talked about in the very beginning of the semester, which is bioremediation. So there are some organisms, as mentioned here, that are acidophiles or alkalinophiles that actually do prefer these particular environments. And so in order to remediate our damage, or in other words, repair the damage that we have done with our acid mining, etc., we can actually use these organisms to help bring back the ecosystems in these regions. So we can introduce these acidophiles that can grow in this very toxic environment, and as a result of their growth, they produce metabolites that become essentially nutrients for the soil to replenish the soil and allow growth for regular or for other neutrophiles, other organisms in that region. And then our salinity terms, we've talked about these in lab already. These are a little bit nuancy, and this is in fact has been taken out of your current edition of the textbook. This is from previous editions, but some terms that are still used. Organisms that grow optimally with very low concentrations of salt, but they can tolerate up to 10%. We call them halotolerant organisms. Organisms that require a high salt concentration, so they grow optimally at about 3 to 5%. These are halophiles. Our skin organisms kind of fall into this category. So our Staphylococcus epidermidis in particular is a halophile. They do have evolutionary adaptations to live in that high salt environment by increasing their own salinity, so they do require that high salt concentration. And then organisms that require a very high concentration, usually above 10% or above, we call them extreme halophiles. These terms should look familiar. We use these terms to kind of characterize bacterial growth in our saline plates in the laboratory. And again, they are a little bit nuanced, so your textbook doesn't really have this distinction anymore. For ecological associations, these are terms I've been referencing sort of since the beginning of this semester, and ones that you can find in any standard general biology or ecology textbook. So these are looking at symbiotic versus or symbiotic relationships between organisms. Symbiosis simply means two organisms living in relation to one another. So these are relationships where at least one of the two members of that relationship requires the other for existence, for survival. This is as opposed to other relationships that are between two free living organisms that don't require each other for existence. So symbiosis, I know that most people kind of have a positive association, like this is something beneficial, but in fact it's not necessarily. There are 3 different categories of symbiotic relationships: mutualistic, commensal, and parasitic. So all of these things mean means something different, right? So symbiotic, again, just simply means two organisms living in relationship with one another where one requires the other for survival. For mutualism, and let's see if I can get my pen to work here. For mutualism, we call these a plus. So these are those beneficial, where one will benefit and the other will benefit. So both organisms benefit in this relationship. A great example of this in microbiology is everything we've been talking about for the first few weeks of the semester, the beneficial flora, normal flora. Those that we've specifically mentioned bring benefit to the host. So these guys can, for example, protect against pathogenic colonization through antimicrobials, etc. So they give us some type of benefit, and at the same time we are providing a host, or we are providing a habitat for them, so we are providing them a benefit as well. So in these relationships, they are beneficial to both. Commensalism, this is a little bit different. This one we will refer to this as plus NA. So one organism will benefit in this relationship at neither cost nor benefit to the other. So one of the two in this relationship is not harmed and is not benefited. The best example, so classically in ecology, I like to give the example of barnacles on a whale, where the barnacles benefit, but the whale is neither harmed nor really helped in that relationship. But that doesn't really help us here. So I think the best example of this one would be a carrier, like that super carrier for coronavirus. So a carrier is, a human or animal that carries an organism, like a coronavirus, which is not an organism but a pathogen, or typhoid Mary, for example, right? An organism that a host that carries a pathogen that is not harmed by that carrier state, but the pathogen definitely benefits in this relationship. It doesn't harm the host, it doesn't benefit the host, but it is benefited by having a habitat there, right? So carrier-pathogen relationship. For parasitism, this would be one will be benefited. One will be benefited at the harm or detriment of the other. And you can already come up with many examples of this, but let's just say our example will be pathogens. So any kind of pathogen would be a parasite, would be benefiting at the cost of the host. So these types of relationships, again, for symbiosis, these refer to relationships between two organisms where one requires the other for existence. For synergism, this is different. This is relationships between two free-living organisms that do not require each other for existence. Well, there you go, interrelation between two free-living organisms. Oftentimes, or in all cases, synergistic relationships will be beneficial, but are not necessary for survival. So this is in contrast to mutualistic symbiosis, right, where these are both mutualistic relationship or beneficial relationships one to the other. But in this case, it is actually required for survival. So if you take away the host, then you actually eliminate the ability for that other organism to survive. This is also called coevolution, right? So where two organisms have evolved over millennia or at least tens of thousands of years, so that the existence of one is reliant upon the existence of the other. Really classically in general biology, mutualistic or coevolution is seen in something like the pollinators and the pollinating plants, where exactly is true. The same exact thing is true. The existence of one is required for the existence of the other. But in synergistic relationships, these are beneficial relationships, but are definitely not necessary for survival. For microorganisms, there are many examples of this. I think one of the most classic ways to think about synergistic relationships is to think of biofilms, or biofilm communities. where, again, these are all free living organisms, all these microorganisms. But that biofilm community, the organisms that are able to produce that glycocalyx layer, certainly improve the survivability, but that biofilm is not necessary for survival for organisms that live within it. It is definitely beneficial, but not necessary. And then antagonism would be also interrelationship between two free living organisms, but in this case, it's detrimental. So synergism and antagonism are similar in that the organisms in these relationships are free living. They don't require each other for survival. But for synergistic relationships, these are beneficial relationships. For antagonistic relationships, these are detrimental. Great examples of where you might see antagonism are things like antimicrobials. We talked about a few of these already. Whoops, that should say that. Like ruderin, we've talked about. That's secreted by Lactobacillus reuteri, that's antimicrobial, or any antibiotic like penicillin. Those are great examples of microbial antagonism. Penicillin, where these are antimicrobials directly secreted from one microorganism to inhibit the growth of other microorganisms in that environment. Oops. This is a picture from your textbook to show all of these things. I like it because it does segregate really nicely between all of these relationships where the organism live in relationship with one another, and at least one of the members of that party require the existence of the other, versus non-symbiotic relationships. These guys are free living. They do not require each other for survival, but definitely have relationships or interactions. This is where you would see synergism versus antagonism. Here on the symbiotic angle, these live with each other, right? Mutualism, commensalism, and parasitism. So with this, you should be able to answer a couple of these questions. The first, differentiate between parasitism and microbial antagonism. And again, there are similarities, right? Parasitism is a plus-minus relationship. Microbial antagonism you can consider as akin to competition. And in competition, we almost always say this is a negative relationship, where even in a scenario where one outcompetes the other for resources, they still are harmed in the sense of requiring the input of energy. So it does cost both parties in a competition relationship when they are competing for resources. So anyway, you can kind of go back and forth between these things. We're talking about parasitism and antagonism, and we'll talk about this a lot more in the lecture, about the benefits or about the similarities and differences between these two things. The second question here is one that you might see on the exam, something similar. E. coli provides humans with vitamin K when colonized in the gut. We've talked about this from our first chapter. This is an example of one of the benefits of normal flora, how it helps improve metabolism, or the provision of nutrients like vitamin K, which is important in blood clotting. What relationship do we have with this bacteria? Well, you can go back to our picture here, or even to the notes that we made here. We already identified one of these categories as being kind of a definition of normal flora, right? And that would be symbiosis mutualism here, or both will benefit in this relationship. We benefit by the provision of vitamin K, and E. coli benefits by having a host. And then this last section is microbial growth. Pause here. So for microbial growth, we are discussing kind of two different types when talking about growth. In fact, the first one is something that we'll discuss a lot more in Chapter 7 on metabolism, which is looking at individual cells and how they grow and expand before the process of cell division. So this is looking at everything from transcription, translation, producing proteins, to understanding enzyme reactions, biochemistry, and specifically looking at biosynthesis, so the development of new cellular components. But once that one cell divides into two, now we're discussing population growth, right? So as soon as you begin to discuss cell division, then you begin to discuss population. Cells divide through the process of binary fission, wherein one cell divides to make two exact copies of itself. This is a type of cloning in essence, right? From one cell to two exact copies with no variation. This is also asexual reproduction. Cells, remember, divide across that transverse plane or the width of the cell to make two copies of itself. This also is what determines its arrangement in cell culture. The process is as follows. This is from your textbook. So this is a cell that is growing and expanding. Again, we'll look at some of the different biochemistry in the laboratory and also in Chapter 7 on biosynthesis and Chapter 8 on replication of DNA. So the young cell expands in size, and right before the process of cell division, we have DNA replication. So the chromosome is replicated, and this is through the process of semi-conservative replication. So you actually don't have an old and new copy. These essentially are the same copies, one to the other. But we'll look at that process in Chapter 8 a little bit more detail. So once you have DNA replication, then you have a septum formed where that cell membrane essentially comes in, invaginates. You have cytoskeletal rearrangement here. You have the segregation of different parts of the cell. So for example, ribosomes will segregate from one side of the cell to the other. And then you have the complete cell division altogether. Now this is sometimes an incomplete division where that septum doesn't completely dissolve, and this is what gives you these different arrangements that we've seen before, streptococci or strepto versus staphylo, etc. The population grows at a very predictable rate in at least a closed system. In nature, this rate is completely varied, actually, depending upon where that organism exists and its interaction with its environment and with other organisms in the environment, but we're going to keep it nice and simple. When a parent cell divides into two daughter cells, we call this the fission cycle. This is also the generation time or the doubling time it takes to double that population or to make that those daughter cells from a parent cell. On average, populations divide, or bacteria divides, every 30 to 60 minutes. 20 is actually quite common too. But there are some examples where organisms can divide very, very rapidly. These are virulence factors, in fact, that organisms that can divide really fast can sometimes establish themselves before the immune system really has a chance to establish its own response. This is seen in, for example, necrotizing fasciitis, that flesh-eating disease where those pathogens have a really fast rate of replication. They also have toxins that they secrete, which is what causes the necrosis of the tissue, but their really fast rate of replication, 8 to 10 minutes every generation, makes that population just explode very rapidly. Some organisms have a much longer generation time. For example, the tuberculin bacterium, Mycobacterium, replicates once every few weeks to months. So for the organism that causes, that's tuberculosis I just mentioned, but leprosy, the other Mycobacterium leprae, divides, it takes a much longer period of time. In fact, you are not cleared for leprosy infection after exposure for, I think, 2 decades because of the slow rate of replication. So you actually can't detect this necessarily at high levels for at least a decade after exposure on some circumstances. So because this is 1 to 2, 2 to 4, this is also considered exponential or logarithmic growth, and we can establish or utilize this population estimation equation to determine the population, the actual quantity of bacteria in a particular region. And you just need to know a few numbers here. So don't be daunted by this equation. It's actually kind of simple. The NF is the total number of cells at a given or final time. Let me see. Okay, so let's say that you were curious about how many organisms are in that hamburger patty, for example, that you left on the counter. If you know the initial number of potential pathogens that are in that sample and you know that pathogen's generation time, so how long it takes for it to divide from one bacteria to two, then you can give a determination for how many organisms are in that beef patty or whatever it is you're analyzing after a given period of time. So to calculate this, let's do an example of this equation. So let's do an example where we're going to do an example with Shigella. Shigella is not super common, but this is a food contaminant. It's a gram-negative Enterobacteriaceae, a member of the same family as E. coli. And this guy has a generation time, so it divides about every 20 minutes. We'll keep it simple. And so we can calculate n if we know how much time has elapsed. And let's just say our initial number of bacteria is 2. So let's just give a ridiculous example that you went to lunch and you had a burger, and the chef in the back was not that hygienic and had a little bit of his own Shigella bacteria from a previous diarrheal infection still on his hands. Whoops. Okay, so we're working through this population estimation equation. And to do so, let's do an example. Let's see if I can pull this up. So if you were calculating or trying to enumerate a population, if you were trying to calculate the number of bacteria in a population after a given period of time, then you can use this exponential growth population estimation equation. We're going to give an example with Shigella, which is a gastroenteric pathogen, causes gastroenteritis. Shigella, there's lots of different species of this, but we'll just say in general, the ID50 of Shigella is about 5,000. So let's say you go out to lunch yesterday, or you went out to lunch yesterday and you got a hamburger, and that worker was not completely sanitary and left maybe 2 of his previous Shigella bacteria on the burger. So the starting number of cells in this case, or the initial amount of bacteria in our example, Ni, would be 2, let's just say. Now Shigella replicates once every 20 minutes. So 20 minutes is the generation time. So there's a couple of things you can do with this number. If we know that every 20 minutes Shigella replicates, then how many generations pass per hour? Well, that's 20, 20, 20, and 20. So 60 minutes divided by 20 is 3 generations per hour, right? So 60 divided by 20 minutes of the generation time makes 3 generations per hour. So if we were to try to figure out how many Shigella bacteria are on that burger that you accidentally left out on your countertop, and you're wondering if it's still safe to eat after maybe 4 hours, then we can use this number to plug into this equation to figure out how many Shigella bacteria are infiltrated into that hamburger. So N is the generation number, so how many generations have passed within that time elapsed. In our example, let's just say there's been 4 hours passed. So again, there's 2 different ways you can calculate this. You can just say we know that there's 3 generations of Shigella bacteria generated per hour, so we can just say 4 hours times 3 generations per hour makes 12 generations total, which is our N. Alternately, if we didn't calculate this number and we just took this initial 20 minutes, we could say in 4 hours, 4 times 6 is 36. Wait, is that right? 4 times 6 is 24. 200, sorry. That's 240 minutes divided by 20 minutes of the generation time makes same thing. 12 generations, which is again our n number. So let's finish populating this equation. So if we had 2 Shigella initially, we multiply it by 2 because our population doubles, right? So this is our doubling number, or exponential. And then n is just how many generations have passed. So n is 12 in this case. And 2 to the 12th is something around 4,000 times 2 makes something around 8,000. That should be squiggly marks. Something like 8,400 bacteria. Now, again, your question is this burger safe to eat? Well, now we just need to go back up and remind ourselves that at 5,000 bacteria, it's infectious in at least half the population. So I would say, well, it's up to you. Okay, so just a reminder again, exponential growth means that we can put this in an exponential representation where the first bacteria that initiates a population or a colony starts with 1, 1 divides into 2, 2 to 4, 4 to 8, 1, 2, 4, 8, 16, 32, etc. It's doubling, right? Every time this doubles, you can represent this as an exponent. So for the first, or for the starting parental population, this is generation number 0, right? There has been no generations developed yet, so 2 to the 0 is 1. After that first population doubling, that is the first generation. You can represent that as 2 to the 1, and that makes 2 cells, and so on and so forth. So if we were to plot this out on a linear scale where time is on the x-axis and number of bacteria on the y-axis, you would see that the number of CFU, and by the way, let me just highlight what this value means. When you see this term CFU, this stands for colony forming unit. It just means it's essentially living cells. Because we know that for each cell that is alive, it can generate an entire colony, right? So it's a colony forming unit. So here we have, down here we have the number of colony forming units just plotted linearly. And you would see in the very beginning, it's really undetectable. 1 to 2, 2 to 4, 4 to 8, 8 to 16, right? It would be way down here. Eventually you get to something like 100,000 to 200,000 to 400,000. So now we can begin to see this huge spike. But very quickly it would go off the scales or off the charts, and it would really be undetectable again on our linear graph. So because of this, we plot a logarithmic scale on our colony forming units y-axis and a linear scale on the x-axis. And in this case, we're doing a log base 2. In our example in the laboratory, we did a log base 10. In either case, you would see this nice linear increase as we're just plotting the log. And remember, for this case, logarithm base 2, it just means like how many generations have passed. So first generation, second generation, third generation are also kind of equivalently represented by these exponents here. And you would just see that like this: first generation, second generation, third generation over time, and just nicely increases linearly. Bacteria, when growing in nature, always grow in biofilm communities. This is almost always the case. This is a picture from your textbook showing the inside of a bandage, I think, after a couple of hours, demonstrating a really nice biofilm community. These biofilms are really complex and very difficult to study in the laboratory, though some scientists do. These microbial ecologists do attempt to evaluate interactions between organisms within these biofilm communities and also how they respond to the environment. But to simplify this, we study bacteria in a laboratory system grown as monocultures or as individual bacteria in these closed or batch systems. Because nutrients are not replenished and waste is not removed in these closed systems, there is a very predictable growth curve that is established. This is your final key concept for this chapter here as well, so just be aware. Here's that population growth curve. We've seen this a few times now. Again, this is a semi-log graph of bacterial growth. In this case, we are looking at the actual count of viable cells or colony-forming units. This is a log base 10, which allows us to distinguish between this initial acclimation stage and this exponential growth phase here. And then here's our hours. Now note that every single bacteria, even from one species to another within the same genre, will have a different population growth curve based on their optimal preferences, right, etc. But let's just understand each of these different stages along the way. So in lag phase, during lag phase, this is our acclimation. So bacteria are adjusting to their new environment. Sometimes you put in a new sugar or new growth substrate, so they have to change their metabolic and gene expression, metabolic pathways and gene expression to adjust for that new media. There is population growth, but it's below a detectable threshold, right? Because again, we're working now in log scale, and this is base 10. So for example, this looks like it's, this might be 1 here. So 10 to the 0, 10 to the 1, 10 to the 2, right? So this would all be all your numbers under 100, or yeah, 1 times 10 to the 0 would be 1. 1 times 10 to the 1 would be 10. 1 times 10 to the 2 would be 100. So all of your organisms would kind of start out way down here as you get from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, right? So all of these things would be way, way, way down here. And then eventually they would pop up in numbers wise so that you begin to see them in this exponential growth phase. Now during this growth phase here, exponential, the population is growing optimally. So this is the optimal growth rate. It's using, expressing, or optimally metabolizing, and has the highest rate of gene expression. And this would be for biosynthesis and for metabolic activity, right? So you would see a very particular biochemical profile expressed here during this exponential growth phase. And because of this, this is the best time for testing, right? So if you wanted to determine characteristics of your bacteria, this is the best window of time for testing. I really hope these slides save. Eventually, the amount of nutrients will be depleted. Bacteria will just continue to grow until there's nothing else to eat. So you have increase or depletion of nutrients. And you have increased waste, which makes bacteria start to die. So you begin to have death here, but you still have bacterial growth. So our stationary phase is where growth is equal to bacterial death. If you have bacteria that are able to sporulate, sporulation would begin here. If you are looking at gene expression, you would begin to see alterations. Ooh. Gene expression. Eventually the nutrient level will deplete too much, so bacteria enter into death phase, and then this remaining period here, period of prolonged, oh my goodness, of decline. Where about 5% of the population remains, essentially scavenging off of the nutrients that are released upon dying bacterial cells nearby. So the importance of understanding the population growth curve and understanding how to control for microorganisms. Remember that exponential phase is where they're metabolizing optimally, having a particular panel of genes expressed, and therefore if we want to try to control for microorganisms or reduce microbial infection in the human, we probably want to target them when those genes or metabolites are optimally expressed. And understanding how to use these in food, for example, fermentation of our food items, we do need to understand how they optimally grow and how to get them to obtain that exponential growth phase so that they can produce those amazing foods for us, and understanding how to do this in the culture, right? Young cultures are better for testing, for biochemical traits, for gram stains, etc. And if you ever were going to industry, you would need to understand how to maintain that stable growth rate. To analyze growth, we've discussed this in laboratory already. There is relative analysis of optical density. This is using a spectrophotometer to do a relative analysis from an uninoculated tube of media into bacterial growth. Again, this is optical density, or OD, that we use here. And for bacteria, it's always an OD of 600 nanometers that is used to evaluate absorbency of bacterial cells to show you transmission or absorbency, and plot these things out to determine those different phases of that population growth. However, if you wanted to do enumeration, so that means that you actually physically wanted to count the number of bacteria in each cell culture, you would do one of these cell counting techniques. Kind of the old tried and true method here is using a particular calibrated microscope slide called a cytometer, wherein you utilize a very specific amount of your cell culture. Usually it's about 10 micrograms or microliters that's put onto this microscope slide. Oftentimes you use a stain so that you can visualize the bacteria, and then under the microscope you simply count the number of bacteria in each of these squares and multiply it by a particular factor. Anymore, we use automated machines called a culture counter or a cytometer. These ones provide the ability for you to add a very specific quantity of your population in a liquid culture. These are filtered through a capillary system that separates out each individual cell from the other. These cells then pass through this counting orifice, which essentially is like it's a light source that has a detector on the other side. And every time there is something that passes through this counting orifice, that transmission is interrupted, and it counts it or detects it as an event or a bacterial cell. So this gives you a nice, very simple way to count the number of cells in any system, but this is undifferentiated, means you can't tell, for example, live versus dead bacteria or any particular characteristic you might be interested in. So if that were the case, that you wanted to differentiate between one type of bacteria from another, for example, let's just go with live versus dead or cytotoxicity, then you would use flow cytometry. And in this case, instead of there just being a light source that's passed through, this is a laser that has very specific excitation wavelengths that it emits. And if there is a fluorophore, let's say, for example, again, I want to do live versus dead, I would stain my cells with a fluorophore that goes into cells that have weakened cell membranes, so the dead cells versus those that have their membranes intact, and that would be my live cells. And each of those fluorophores has a different excitation and emission pattern. And so as these bacterial cells are passing through, the laser will excite them, and whatever the fluorophore they have will then emit a very specific spectrum that is then detected. So this not only counts how many events or how many cells are passing through, but it also tells you what their fluorescence pattern is, which tells you something about the characteristic of the bacteria. And that's it for these sets of slides.